Hello viewers, welcome to Biodrunks. For your reference, I'll be providing notes, PDF link in the description.
Let's start our video with a very important structure in the cell, that is plasma membrane. Okay, so let us know each and everything about plasma membrane in detail. So this term plasma membrane is being coined by Nageli and Karner in 1855. Okay, and the term plasma lamella was given by J.Q. Pauli in 1931. Now, what happens is, why is the cell needing a plasma membrane? For example, this is your house. Okay. So to protect your house, you have a boundary around. If you are not having a boundary, what will happen? The outsiders will come inside without any permission. Now, inside, for example, if you are having kids, they are going to go out, isn't it? So this boundary, it protects your house. What does it do? It protects your house. In a similar fashion, to maintain the integrity of the cell, it is surrounded by a membrane which is called as a plasma membrane. Okay, so I think this is very clear why. Now let's learn in deep about why is it so important. See, what does plasma membrane do is it limits the boundary of the cell, because there is a major difference between the internal cell and the external environment. Means whatever the environment is inside the cell, this is the environment inside the cell, which is quite different from the environment that is outside. Means this feature. Okay, for example, this one cell, this one cell. So this region is called an external environment. External environment. And whatever the environment you are having inside the cell, that is called as internal environment. So you have to separate both of them. There are a lot of differences in that. Okay. And not only that, you also have to confine your cell organelles which are present inside. Okay. For example, there are several organelles that are present inside. If you don't have a boundary, what will happen? You cannot differentiate it as a cell. It might go here to there. So it would be a random exchange and there won't be any stability. And the plasma membrane, okay, it is selectively permeable. This is a very important point, that plasma membrane is selectively permeable. Selectively permeable means it allows only a few things to enter the environment or enter the cell and go out of the cell. Okay. Now, why does it do? So that it can maintain the concentration of electrolytes. If, for example, if you have any electrolyte concentration which is more inside and less outside, okay, so it might lead to your cell signaling problems. Okay, there are several issues which have been faced if you have low and high concentration of electrolytes, biomolecules also. Okay, so to maintain that concentration, it is selectively permeable and it does not allow everything to go inside. So its passage is like gases and then the small molecules and then the larger molecules. Okay. So that is how it allows the things to go in and out of the cell. So according to the permeability, let me write it. So it's like gases and then water, and then you have small molecules, and then you have large molecules, and at the end you have charged ions or movement. Might be a positively charged or a negatively charged. Okay, so this is on the basis of permeability. So this is a very important question being asked in most of the entrance exams. Let's go to the next point. That is, what is plasma membrane made up of? Okay, now plasma membrane is made up of long chain of fatty acids with polar groups attached to them. Okay, and it is a bipolar structure with hydrophilic and hydrophobic tail. I'll be explaining each and every point in detail. Okay, and always remember that hydrophobicity plays a very important role in the structure and function of the cell. Now, how can we know, like, how does a cell look? So, Singer and Nicholson in early 1970s or 1972, they stated a model called as fluid mosaic model. This model helps us to understand the cell. Let us understand it. So how does a cell look like? Okay, fluid mosaic model. Fluid mosaic model. Now, the cell has, this is the head, the cell plasma membrane. Okay, and these are the tails. Okay, these are tails. So it's a bipolar structure. So this is how it is. Okay, so it's very easy to draw fluid mosaic model. You can draw it this way. Now, this is the plasma membrane, the main structure. This is called as phospholipid bilayer. Okay. Now you have integral proteins here. These are called as integral proteins. And there are some peripheral proteins also. Let us enlarge it a bit more to understand it properly. Okay. So this is a phospholipid bilayer. Now I'll explain how does this phospholipid bilayer look. This is how it is. Okay. So here I've mentioned the basic structures. Let's first understand each one by one. Let's label it. So this is called as an peripheral protein. Okay, peripheral protein. This is your integral protein, means because it's inside, integral protein. Okay, and this is a phospholipid bilayer. Okay, so this is a phospholipid bilayer, and this model is fluid mosaic model. Okay, now this phospholipid bilayer, let us draw it like this. Okay, now what each structure is? This is your hydrophilic head. So when I say hydrophilic head, it means water liking. Okay, they can stay in the water. These are hydrophobic tail. So when I say hydrophobic tail, that means they are water hating. Hydrophobic means water hating. Okay, so this is hydrophilic head and a hydrophobic tail. So this is a phospholipid bilayer. So I'll be doing one video about phospholipids or lipids in short, okay, where I'll be explaining each and every part in detail. So till now you can understand that this is a phospholipid bilayer. Now what is it? What does it constitute? So this head part has alcohol, okay. Then you have a phosphate moiety, phosphate, and to this you have glycerol. Okay, and then you have in the tail part, that is your fatty acid tail. Okay, this is your fatty acid to fatty acid tails. So this is how your structure looks like. Okay, this is the phospholipid bilayer. Now, always remember these are the most important things in a cell. Okay, this hydrophobicity is very important because they don't like water, so they are inside. Okay, if you can see, they are inside, and these are on the outer surfaces. This is outside the cell, this is inside the cell. Okay, so now this is the basic layer of phospholipids. Okay, hope so it's very easy, and you might have understood what is a fluid mosaic model and how does your plasma membrane look like.
This was all about today's video. Hope you liked it. For more such interesting video, please like, share and subscribe to BioDrunk. Let's meet in the next video. Bye-bye.